Hello, Jeff Zwerink here, and welcome to Give and Take, the segment of our show where we explore important scientific ideas and ask how do they relate to the truth of the Christian faith. Today I'm joined by Anjeanette Roberts, and we're going to explore just how complete is the human genome. AJ, it's good to have you here today.
Hi, Jeff.
So just kind of a little bit of background. Give us, what is the genome when we're talking about that? What is it that we're talking about, and why is it important?
So a genome is the DNA in an organism's cell. Most of it's in your nucleus. There's a little bit associated with the mitochondria, the powerhouse.
Okay.
But it's important because the genome is what encodes all of the proteins and all of the regulatory RNAs that help a cell function. So any protein in your body is encoded in your genome. It's a blueprint for life.
Yeah, that's what it sounded like. This is not trivial stuff. This is kind of the foundation of life, if you will. So the idea that we're understanding how the genome works is pretty important, is it not?
It is, yes. And for humans, we have 23 chromosomes in pairs.
Okay.
So if you've heard of 23andMe, that's referring to our genome in 23 chromosome pairs.
Okay, so it's wrapped up in 23 different pairs. Is the same genome in all 23 chromosomes, or there's different parts of it in all 23 there?
It's different parts in all 23. I mean, there are some repetitive sequences, but for the most part, each chromosome is unique.
Okay, so there's been a lot of talk, a lot of press lately about how we've got the human genome mapped out and other species mapped out. This is a big deal. So I guess the question is, how completely do we have them mapped out? Do we have them for lots of organisms and humanity?
Well, it's a great question because there's a little bit of ambiguity in what you mean when you ask the question mapped out. So we have many different organisms, all the way from bacteria, viruses, up to humans mapped out. But we don't have any complete genomes for any organisms above the level of bacteria. So we have some complete bacterial genomes, we have some complete virus genomes, but all other animal genomes are incomplete to various degrees, so including ours.
So is that just because of the complexity of the human genome, or is it the size of it? Or I kind of give a little bit of background. What's the difference there? Why can we get the bacteria and not the human genome?
Well, so a bacteria has a single circular DNA molecule that's a bacterial chromosome, and so it's pretty easily accessible. But yes, for the human genome, we have what's called areas of heterochromatin, and that's areas of our DNA that are tightly bound to proteins that are really hard to sort of get them unbound and to get access to those. So there's a lot of areas in our genome that are heterochromatic that we can't quite access or sequence through.
So it's more like it's just bound to something, so we can't, in essence, break it apart to read what's there. Is that the idea?
Yeah, that's part of it. We can't either break it apart to read what's there or break it apart to capture it. There's also areas that are highly repetitious, and so the way that we capture most of the sequences from genomes makes it really hard to see how long repetitious sequences go on, because we capture just snapshots of the repetition, and so we don't have all of the repetitive sequences of the chromosomes mapped out either. So
is that because just in the repetition it's hard to know how far you've gone through there, or
yes,
so you can't just lay it down and kind of read it off letter by letter, if you will?
For the most part, we can't, because most of the genome was sequenced in the earlier days by either capturing fragments and sequencing the fragments that were captured,
okay,
or by sort of doing a shotgun approach, and it's called that because you basically try to capture really small fragments of the genome and amplify those, but you're working with such small fragments that you don't have long reads, and so the long reads are what tell you how long repetitious sequences are. We can now capture those through different technologies, but we couldn't in the days that they were assembling the human genome the first time.
So it sounds like it's just pretty challenging work. I don't want to gloss over how remarkable it is what we've been able to do.
Absolutely.
But the fact that it's incomplete is also a significant thing to be aware of.
I believe it's really important that we're aware that it's incomplete. And as research goes on and we start to sequence into those difficult areas, not only are we finding genes that are in those difficult areas that we wouldn't have expected to be there necessarily, but we're also finding stretches of the genome that aren't part of the actual reference genome.
So by the reference genome, presumably we've put together this thing that says, okay, this is to the best extent what we know, and then as we get new stuff, we say, okay, where does that fit in there? You're saying that we're finding in certain people that there are stretches that aren't in the reference genome.
That's correct. There's a recent publication earlier this year in 2019 that suggests that they captured 60 million base pairs of non-repetitive sequence that's unique in the human genome that's not present in the reference genome.
I mean, that's not, I mean, the human genome's like 3 billion-ish, correct?
3.2
Somewhere in that
billion.
range? Okay, so the 60 million is not an insignificant part of that.
It doesn't seem insignificant, especially when you realize that the new sequences that we're able to sort of unlock, we're finding genes there.
That is pretty remarkable. So kind of the bottom line question for me is, I think this is great research. It's fascinating that we've gotten to this point. It's a remarkable accomplishment. Why is it important that we understand that it's not complete?
Well, there's the comparisons that we make to other organisms and the inferences that we build on those comparisons. But more important than that, I think, is that we now have the ability through CRISPR-Cas9 gene editing technology to edit a genome. And there are individuals, a scientist in China that actually went in and edited embryos. So he changed the human genome of unborn baby girls that were born live. There's at least 2, possibly 3, that had this genetic manipulation done. But if we don't know what 8 to 10% of the human genome is, then we shouldn't be targeting it to select changes when we're not really sure if we're only targeting one site. Because that's one of the major problems with that technology is you want to be sure you're hitting only what you're targeting and you're not getting off-target hits.
So in some sense, there's a little bit of hubris when we say, okay, we can go in and do X, Y, or Z when we don't know what the genome is.
Exactly.
And kind of, coupled in with that is that by not knowing everything in the genome, we can actually do damage that we're just unaware of what we're doing.
Right, and it may not be immediate damage that prevents whatever you're trying to accomplish from ever coming to fruition, but it may be damage that doesn't materialize until later on in the organism's life.
Yeah, it just seems like that's a pretty consequential thing, and especially it's a place where as Christians, or where we have how we view humanity is really important there, and that kind of ties it back to, as Christians, it seems like we have a pretty important voice in this discussion.
I think we absolutely should, and I would actually say that most scientists out there are outraged that this type of research has actually been implemented because they see that it's hubris sort of gone amok.
Well, thanks very much, AJ. I appreciate your comments.
You bet.
We hear a lot of talk about how we've mapped out the human genome, and now as we can start to apply it to technologies where we can edit the genome to get rid of diseases or enhance humanity, we ought to just apply a little bit of appropriate caution there, recognizing that we don't have a complete map of the human genome. I'd encourage you to go check out reasons.org and look for AJ's recent blog on this. If you search for a post-genomic era, that will take you to that blog to help you understand the limitations of what we know, but also how as Christians we have an important and vital voice to articulate why are humans valuable and how do we do this kind of research so that we're actually advancing humanity based on our value in God's sight, as opposed to doing things that may cause a lot of damage.